Hi, my name is Madison Ross. It is May 21st, and I will be doing my presentation today on the anatomy and physiology of the retina and how it connects to our central nervous system. So this picture here is just a brief anatomy overview, so we have a good idea of our entire eye going into some physiology within the retina. So we'll start at the front where we have our anterior chamber, which will hold our aqueous humor, which is a watery liquid that will fill the space between the cornea and the lens and helps to maintain that front shape of the eye. We have our iris, which sits just in front of the lens, and will change size according to the pupil, and also this is what's going to give you your eye color. And then we have that pupil, which is going to be the hole in the center of the iris, which will open more to let more light in and will close more in super bright light to let less light into the eye. And then as we move further back, we have our fovea, which is actually going to be a section of our retina, which we will talk about a little bit more. Directly behind that pupil, that contains a large number of color-sensitive cells, and this is why you should look directly at an object to see it most clearly, because we have this section in the back of our eye that when the light hits correctly and everything lines up will allow us to see the best pictures. But today we really are going to focus mainly on the retina. So within the retina, or actually the innermost layer of our eye, as you can see back here in the peripheral retina, it's made up of hundreds of millions of neurons, and many of which are photoreceptors. Now, those photoreceptors are what respond to our light signals and are going to be distinguishable from other cells because they are made up of densely packed membranes and actually contain photopigmented rhodopsin. Now, the rhodopsin pigment contains sensory proteins that convert light into an electrical signal. And if you know anything about our nervous system and how they communicate, you'd know that things communicate through electrical impulses in that area of our body. So next we have our physiology and our visual pathway, just a little bit more information here. So the retina is going to be broken down into 3 layers of cells. So it's going to start with those photoreceptor cells that we talked about. We then have our bipolar cells, which you can see here in the middle. And then after that we will have our ganglion cells. Now the layer of photoreceptors that contain that rhodopsin actually will undergo the process of phototransduction. Now that phototransduction is the process that breaks our light photons into those electrical impulses and allows them to move further through the system to move those impulses. So the process starts with the photoreceptors, and then once it's turned into an electrical impulse, it will move to the bipolar cells, and then it will move after that into the ganglion cells. There are different kinds of photoreceptors. We have both our rods and our cones, which you can see here in the middle, and are named rightfully so, as the cones have more of a cone shape, and our rods are a little bit longer and leaner. The rods are going to be responsible for interpreting size, shape, how bright an object is, while the cone focuses on fine details of an object and their color. And then we have our axons of the ganglia cells, which actually will compose the optic nerve, which in this picture would be leaving right through back here, which will relay the impulses to the first visual relay in the brain. Just a couple more things in that visual pathway. We have our horizontal cells, which help to receive input from multiple photoreceptors at a time, as you can see down here in this image. You can see them between the bipolar cells and the photoreceptors. They will actually combine signals to make adjustments to them before they move on to the bipolar cells. And they help to regulate the activity of the photoreceptors themselves as they start our pathway of neural communication. Then we have our amacrine cells, which you can see down underneath our bipolar cells, between them and the ganglion cells. Now, they help to receive signals from the bipolar cells and they help with regulation and integration of the activity in both bipolar cells and the ganglion cells. They are intrinsic intraneurons that are described as the most diverse class of neurons. Something that I found very interesting that I'd like to talk about in this section is you can see, and you kind of saw back in the other picture, that as we talked about how the light moves and how the neural communication moves, we started at the photoreceptors, which seems to be in the back of the eye, and then we talked about the communication moving forward through all of those different steps to the front of the ganglion, which seemed to be in the front of the eye, which doesn't seem to make too much sense if you are thinking about it moving in the way of the direction of the light, but they are actually going to be opposite of each other. So as you can see on here, I have two different sets of arrows, one representing the direction of neural communication, which we talked about on the last slide, being how it starts in the photoreceptors, with that phototransduction being turned into the electrical impulse, and then everything else will move down that line. But in reality, the direction of the light will actually hit the ganglion cells first and then move back in the opposite direction. This picture is going to talk about our visual stimuli response in the central nervous system. So now we are out of that internal retina and we have moved backwards into the brain. So once our optic nerve leaves through the optic disc, which is a small hole in the back of the eye, which allows for that nerve to leave, they will converge at the optic chiasm, which you can see here. Here, multiple axons will undergo decussation, which is the crossing of nerve fibers to allow the crossed processing and interpreting the visual signals on opposite sides of the nervous system. So as you can see here, this red line on the inside, which will act as the nerve coming in from this side, actually crosses over and will be read and interpreted on the other side of the brain coming through into this other optic tract, and so forth. Here, this internal green one will actually cross over, meeting up with the peripheral nerve that travels on the temporal side of the outside of this eye and will cross over here. And this happens so one eye is not registering on one half of the brain. It allows them to cross over and it will actually mix the signals so you can get a better picture. Like I just mentioned, as it says here on the slide, this will allow for visual input from two halves of each separate retina to combine into the same portion of said visual field that we are interpreting. The axons continue straight onto the optic tract from the temporal side of the retina. Like I mentioned, this outside one here will not cross over in the middle, same with the red one on the outside here. There's no need for the axons to cross as they come through, and the optic nerve actually terminates here on our lateral geniculate nucleus. But in our next slide, we're going to talk about after it passes all the way back through here into our primary visual cortex, where those images and those electrical impulses actually start to become real images and pictures that we interpret and register and we see on the daily. So just to finish out our brain pathway of how things are registered, the final step in that neural pathway of our visual stimulus is when the electrical impulses hit our primary visual cortex, as you saw in that last picture, is all the way in the back of that tract, and the brain can now start to put back those images together. We have our V1 portion of the visual cortex that lies posterior in the section of the occipital lobe, which is the first part of the visual cortex to start piecing these images together. And it's going to use those peripheral images, so those images that do not cross at the optic chiasm first. They start getting processed in the anterior part of our V1, and they're processed first. Once we hit the V2, the cortex starts to be able to process more complex information, shapes, colors, depth, width, things like that. And our V2 is actually split into 4 quadrants that help us put together a picture of this visual field. So we have more information and things like that. And this is how our visual pathway helps to function in our central nervous system and how we register those things. These are my references, and I hope you enjoyed, and I hope you felt like you learned something.